So welcome everybody. Welcome to our Jameel Clinic seminar series. Today we have the chance to have a great speaker. This is Vivek Farias. Professor Farias is the Patrick J. McGovern Professor at the MIT Sloan School of Management, and he's also affiliated with the Operations Management Group and the Operations Research Center. His research is focused on optimization and inference in large-scale stochastic systems. He serves on editorial boards of management science, where he's the editor of the data science area, on operations research, and the Informs journal on optimization. He received his PhD in electrical engineering at Stanford University. And outside of academia, he's been on the news lately because he's well known for being the co-founder and CTO of Select, which was acquired by Nike, and serves also on several technology startups, advisory boards, and has worked in various capacities in quantitative finance and private equity. I know that sounds a lot of finance, and today we're more on the healthcare, but this is to show a little bit of how the work of Professor Farias spans in different domains. The way we have the event today is, I think we have the full session with Vivek. We're not going to have any clinical collaborator, but if any question, please feel free to interrupt or text, chat, raise hands. We'll try to accommodate as the time permits. And yes, we need to finish this at 5, so make sure that we're on time. And I think without further ado, and if no other question, Vivek, the stage is yours. Thank you very much for joining us today.
Hey, thanks, thanks, Ignacio. You can hear me fine, right?
Absolutely. Thank you, Vivek.
Perfect. Great. So, thanks for having me. Let me just say by kind of way of introduction, in terms of the players over here, I'm going to be talking about work primarily with a former student of mine, Andy Lee. Andy is a professor in CMU, in OR and ACO over there. And Tianyi Pang is a current PhD student in Lids in ECS. Ignacio talked about clinical folks. I was a little sloppy, and I probably should have invited them. But I'm talking about several projects over here that wrap up under the same kind of umbrella. But in terms of the clinical work, I'll be sort of talking about some work that I've done with Omid Farrokhzad's group at the Brigham, along with Bob Langer. And that'll be one piece. And then I'll talk also about some work that we've been doing. The work with Omid and Bob is published. And there's 2 papers which I'll share. One was published late last year. And then there's ongoing work with Steve Carr's group at the Broad. And so I'll kind of touch on those things. That's kind of the objective over here. But really what I'm going to try and do today, is talk about a very simple idea at the end of the day. It's a simple idea of really optimal recovery in tensor data. I'll try to make clear what I mean by that in a second. And the high level sort of story over here is that what we'll be able to do is, number one, from an algorithmic or theoretical perspective, present a really simple algorithm that actually is surprisingly the first optimal algorithm for a problem where traditional convex relaxations apparently are very hard, don't work well. And so that will be sort of a theoretical sort of nugget that'll sort of be interesting, especially given how simple this algorithm is going to be. But then second, I'm going to try and convince you that this algorithm is, from an engineering perspective, actually quite useful. It's useful by itself, and it's also useful as a module, as part of sort of a larger sort of machine learning pipeline in some of these biological applications. So that's kind of the high level. Feel free to interrupt at any point in time. The more interactive, the better. But let me get going here. So at a very high level, this is like a 100,000-foot level, right? What am I sort of thinking about over here? So I'm sort of thinking about settings, and I'll make this more precise in just a bit, right? But I'm thinking about settings where we have relatively small amounts of very high dimensional data. So this happens all of the time. Right. So for instance, Steve Carr's lab does sort of deep proteomics, which I can talk about in a second, where in some sense, the ambient dimensionality of the data is very, very, very large, right? But you're fundamentally limited by the number of like biological samples you have, which is maybe in like the low hundreds at best, right? Or something like that. The same thing is going to come up in sort of like a diagnostic setting, which I'll talk about separately. Yet another example, which I think I'm going to use as a level set because it's probably a reasonably well-known data set to a lot of people over here, is CMAP, right? So, CMAP probably kicked off really all the energy and excitement with sort of like, single cell analysis. But if you kind of think about CMAP as a data set, again, it's got this feature that, it's very, very high, high dimensional data, but with a relatively small number of data points, right? Like the number of if you know this data set, the number of perturbagen cell pairs in the data set, the number of experiments, as it were, is sort of relatively sparse compared to, the product of perturbagens in cells. So on the one hand, then we've got very high dimensional data and small amounts of this very high dimensional data. And given this, we want to ask on the flip side, typically very refined questions. Okay, so for instance, for the CAR group, that's sort of looking at like post-translational modification, right? They're asking questions about, ubiquitination at a specific site, and this is a very, very, specific question. It's a very, very refined question. In the more common, let's say, CMAP data set, right? You're potentially interested in predicting the impact of, a perturbagen on expression on a particular gene, a particular group of genes in a specific type of cell. So really, we're asking very refined questions at the end of the day, right? And so at some level, these two things are a little far apart from each other. And so what I'm going to talk about, okay, is looking at these sorts of problems through the lens of tensor recovery. And as promised, I'm going to do two things. I'm going to give you this really simple, embarrassingly simple algorithm, okay, for this well-studied problem, right? And then I'm going to present a bunch of applications of it, right? To one, sort of this sort of deep proteomics thing, two, to CMAP, three, to kind of diagnostics from sort of a novel sensor. Okay, so that's kind of the roadmap. Okay, so just to kind of like set things up, right? Like just to kind of start really simple, and we'll make it more and more complicated as we go along, right? Let's think about sort of this ubiquitination thing that I was talking about. Okay, so what is this, right? At the end of the day, we have tissue samples, okay, from a bunch of folks, right? In this particular case, okay, these samples are drawn from healthy tissue and sort of cancerous tissue on the same patient for a whole bunch of different patients, right? So basically, you've got this list of samples, right? And on this list of samples, you collect a whole bunch of different data, a whole bunch of multi-omic data. Let's say we're specifically interested in ubiquitination, which is a type of post-translational modification. Okay, so what do you want to get a hold of, right? What is it that we really want? What we really want is, hey, we want to understand for each of these samples across the different sites, right, that you can have, and there's lots of these sites, right? To a first order, was there ubiquitination or not? What was the extent of ubiquitination? Okay, and so as a picture, it's this grayscale matrix that you might want to recover. And I've drawn this as a square matrix, which is a little bit misleading, right? It's actually more of it's sort of like a fat matrix. Okay, but I just for convenience, because I'm lazy, I've drawn it as a square matrix. Okay, so I care to recover this grayscale picture. And what do I actually have? What I have to do this is basically mass spec data, right? At the end of the day. Okay, so at the end of the day, I have mass spec data, and I've deliberately drawn this mass spec data in sort of like a digital kind of way, right? It's like almost a black and white matrix over here. And this is to sort of say that the mass spec data is extremely noisy. That's one thing, right? And two, it's such that you may just simply not have this data on samples. So, you'll notice in the big matrix on the left, there are entire rows that are missing, right? So what do we care about doing at the end of the day? What we care about doing at the end of the day is going from the data that we have on the left the ubiquitination, the true kind of ubiquitination, the true kind of thing of what's going on in these samples on the right. Right? That's really what we're maybe trying to recover. So if we sort of think about this for a second, right? Fundamentally, sparse data is going to limit recovery, right? If you sort of tried saying, listen, I want to recover this by assuming, let's say, for instance, that this matrix was a low rank matrix, one way of sort of thinking about the complexity of the model you can pick is how large can that rank be? And roughly the rank is going to correspond to the rank you can pick. It's sort of like the average number of observations per row. Yeah, that's something, or average number of observations per the smaller dimension, right? That's really what you're going to say. And here, of course, the story is even worse because there's entire sort of rows missing. So it's probably not hard to kind of motivate that what you want to do here. This is kind of crazy to just look at this one slice by itself and denoise it, right? The obvious thing that hopefully all of us would want to do is to say, hey, we don't just have data on ubiquitination, we have all sorts of data, right? That we can kind of line up. And so presumably all of that data will help, right, in learning about ubiquitination. Okay, so hopefully I've not said anything particularly surprising so far, right? Hopefully this is exactly what you would want to do. You'd say, hey, look, there's lots of downstream biological applications to actually understanding ubiquitination on these samples because of the data collection process, right? We rely on mass spec at the end of the day. That's something really, really hard to do. And so denoising or imputation, that's something that's very hard. We'd like to solve for. And we just looked at the ubiquitination by itself. That's kind of pointless. We have all of this other data. We have all this multi-omic data. Hopefully we're actually able to use it, right? And so hopefully what you're thinking is a picture like this, okay? Hopefully what you're saying is, listen, if I thought about this conceptually, right? I have a bunch of information for each tissue sample. So in addition to kind of like the ubiquital proteomic information, I have information potentially on all sorts of other PTM, right? I could have, phosphoproteomic information. I could have acyloproteomic information. In fact, we do, right? In addition to this proteomic information, this PTM information, I could have just global proteomic information, right? Like what's the expression of proteins? I could have, gene expression information, right? All from sort of the same, the same sample, right? And so if I kind of line this up, the picture that you should have, right, is on the one hand, we have sites, right? So this could be, the gene mapping to sort of a protein, mapping to a whole bunch of, amino acids on that protein, right? And you can line that up. So that's the sites. I have a number of samples. These correspond to our tissue samples, right? And each of these slices is like, the slice I was showing you over here, right? Each of these slices looks something like this, right? And so I have a slice maybe for gene expression, global proteomics, ubiquitination. And what I want to do, right, at the end of the day, conceptually, is use data across all these slices to really kind of get a nice picture of one of the slices. Okay? So that's what I'd like to do here. Okay, so that's one example, right? But the thing is, this example is all over the place. All right? So let me give you another quick example of the same sort of picture, right? So here's another example of the same sort of picture. And this is CMAP. Okay, so the let's call it the mother of single cell experiments, right? So what is this CMAP data set, right? The CMAP data set, many of you are potentially familiar with this data set, but in case you aren't, right? You have a whole bunch of different cell types, right? And each of these cell types are treated with a perturbagen. Now, every perturbagen or a drug, right? In simple terms, right? So this perturbagen, a given perturbagen isn't applied to all cells, right? Because we don't have the ability to run so many experiments. But in principle, right? I basically take a cell type, I treat it with a perturbagen, and then I kind of observe, gene expression in CMAP. It's about 1,000 genes that are viewed as kind of like, landmark sort of genes, right? Where you're actually measuring gene expression. So now let's just think about this for a second, right? Like, what are we looking at? If you think about this data set, one way of organizing this data set is to say, hey, look, for a given perturbagen, whatever that perturbagen is, right? We'll look at concrete examples later. But for a given perturbagen, I actually have a set of experiments that run with that perturbagen, right? Like I've tried that perturbagen on maybe, cells from like, from the skin, or some other type of cell, or some type of tumor cell, or whatever it is, right? So I have this whole library of cells. I've taken that perturbagen. I've basically tried that perturbagen out on some subset of that library of cells, and I've got gene expression. And then I do this for another perturbagen and another perturbagen and so forth. Now, of course, I can't do every perturbagen cell pair. It's going to take me forever. It's going to be too expensive, right? And so the whole point is, can I, in effect, right, understand the impact of a specific perturbagen on expression in a specific cell type when you've actually not done that experiment, right? And that's, again, the same thing as the previous slide, right? Essentially, what we want to do is hopefully learn across the slices over here, right? We want to learn across these perturbagens. So the point being that as we get more and more perturbagens, any individual perturbagen, right, we can sort of impute better, we can predict better. Okay, so that's example number 2. And I'll come back to this example later, okay? I'll come back to the earlier example later as well. Okay, here's a third example, right? And this is from work that we were doing with Omid and Bob, right? So Omid's lab built a specific type of sensor, okay? What that sensor does is it consists of a bunch of different beads, a bunch of different nanoparticles, and each nanoparticle is very carefully engineered, right? For zeta potential, hydrophobicity, hydrophilicity, whatever it is, right? They're experts at that. So they've engineered a whole bunch of different nanoparticles, right? And what you do is you expose these nanoparticles to plasma, and then you do, the corona forms on the nanoparticle, and you wash the corona off, and you do mass spec, right? And so essentially for any given sample, right? So let's say for plasma, for example, it doesn't have to be plasma, but let's say plasma from a given patient, right? You basically, across each of these nanoparticles, which are distinct, you'd actually be able to get sort of, what is the composition of the corona, the protein corona that forms on that nanoparticle, okay? And the hope is that when you look at something like this, okay, that's actually really useful as a diagnostic tool. Later on, you'll see that we successfully use this as a diagnostic tool in a large study for non-small cell lung cancer, right? But that's the idea, right? Like, so for every patient, now in this case, each patient is a slice. And what are we observing on the patient? We're observing essentially this 2D array, which looks like kind of the protein spectrum, but across different nanoparticles that have kind of been engineered differently, from each other. Okay, so same story, right? At the end of the day, and what we're hoping for over here is as we get more and more patients, obviously, we want to be able to denoise any individual patient better, right? The hope being that downstream for prediction, that's actually a good thing to do. All right. Okay, so 3 examples, right? All basically asking for the same thing. So here's what the rest of the talk is going to be about. Okay, the rest of the talk is going to be about how do we take these sorts of problems, right? And is there something useful we can do to answering the imputation or denoising problems implicit in the questions I just asked, okay? And so what's coming for the rest of this talk, okay, is the following. First, I'm going to say a little bit about algorithms and theory, okay? Just given the crowd, right? So I'm going to say a little bit of algorithms and theory. And so in particular, I'm going to actually be talking about recovery of 3 tensors, which is really what we're seeing over here, right? And what you'll see, or what I'll make the point of, is there's actually a lot of work in this area. And there's actually a big gap between sort of, what's achievable by existing algorithms and lower bounds. What we're going to do is we're going to close that gap for 3-dimensional tensors. Okay, so we'll give an efficient algorithm that closes that gap for 3-dimensional tensors. And these 3-dimensional tensors are really important, right? One way of sort of thinking about it is, in 1 dimension, you have like compressed sensing. In 2 dimensions, you have matrix recovery, which is actually a distinct set of tools. So it stands to reason that maybe 3 dimensions should also be different, right? And indeed, you see that it behaves somewhat differently, okay? And that's kind of the gap we're going to close with a really simple algorithm. What's coming in terms of applications is the following. What I'm going to show you is I'm going to get into each of those things, all right? So what I'm going to show you is for the proteomic data, okay, we're actually able to kind of predict site-specific ubiquitination with an AUC of about 0.9. Okay, and this is actually quite high. So that's that first example that I talked about, the CMAP, the proteomics example, right? On CMAP data, I'm going to show you that we can sort of estimate treatment effects. And so what is the treatment effect here? It's sort of the difference between gene expression under the perturbagen and gene expression, for that same cell under sort of a control, okay, which is basically DMSO. And so what we're going to show over there is actually we're able to recover these treatment effects quite nicely with a pretty big jump over some state-of-the-art baselines that are like barely a year old. Okay, so it's almost a doubling, in fact, of that baseline. And so I'll talk about that. And then for the sensor data, this is the stuff with Bob and Omid. We've done a non-small cell lung cancer study that I mentioned with this new sensor, and it's a study with about 1,000 patients, where we got sort of an AUC for sort of early diagnostics for lung cancer and the AUC was about 0.91. Okay, we did a second paper so I'm not going to talk so much about that because that paper is in Nature Communications as of last year, and I'll give a link to it. You can just go read it, right? The second thing is later last year in 21, we did an Alzheimer's study, which was much smaller. So, take the number here with a grain of salt, but because the study's smaller, but again, same technique, and you get sort of a pretty high AUC, in doing this. And as I said, I'm not going to talk about these last two simply because the papers exist and you can go look them up. Okay. So that's kind of the plan. That's what's coming, right? In some sense, you can kind of stop now, right? And that's really what the talk's about. Let me pause here in case there's any questions.
Good stuff. Thank you, Vivek. Yeah, it's coming well. I don't know if there's any questions.
Okay, I'll take silence as a no. All right. And what I'm going to do next is, as I said, theory first, right? So I'm going to do a little bit of math. All right, it's very simple math. All right. Well, at least what I'm presenting here is relatively simple. Okay, so I want to get the idea across, right? And so it's not a complicated idea. So stop me at any time because everything I'm talking about over here should be relatively like follow a boat. Okay, so let's try and abstract away what I've been talking about, right? So we're talking about sort of recovering a tensor. So what I want to do for starters is for now, just think about a tensor as a multi-way array. Okay, so and because there are 3 tensors, right? Let's think about it as a collection of matrices. Okay, and I'm going to look at the dimensions as m by m by n. And I'm doing m by m, I'm making it square just for convenience. It doesn't have to be a square, it could be a rectangular matrix. There's no problem. In fact, most of these matrices are rectangular. So the m by m is just for convenience. I don't want to track too many things. The n is, how many of these slices do you actually have? Okay, so m by m slices repeated n times, right? And so I'm using the subscript p or superscript p for perturbagen. Okay, so as you think about this, think about the CMAP story. Okay, any individual slice is, let's say, cells across genes, right? That's any individual slice, cells across genes. And then going into the page, you have cells across genes for a whole bunch of perturbagens indexed by p. Okay, so that's sort of what we have, right? That's the true tensor, so to speak. And we don't have the true tensor. So what do we observe? I'm going to say what we observe is a noisy version of this tensor, right? And so in particular, entry-wise, I observe MIJP, right? So an entry of M, plus independent zero mean noise. Not identically distributed, obviously, but independent zero mean noise. Now, as you look at this model, you might be thinking to yourself, hey, wait a second, weren't you talking about like imputation earlier, where things are missing? Well, it's sort of easy to see that there's this sort of old trick, and the trick goes back to like Montanari and O. It's like a 13-year-old trick, where you could sort of think about this additive model equivalently as a missing data model. Okay, so there's a way of defining the epsilons so that equivalently we're looking at entire entries being missing. All right. So what I observe is the true tensor plus noise. All right. So M is the true tensor. The noisy version is X. All right. And so what's the problem that we're after? So the problem that we're after is I get X, boldface X is my data. Okay, that's my input, right? And I want to construct an estimator for every individual slice, right? So for any individual perturbagen P, right? Notice that there's a P over there in the in the third mathematical line, the third equation over here. Right? So for any individual perturbagen, my estimator is going to take in all of the data, the entire tensor, and spit out an estimate of that slice. And what I care about is the error in recovering any individual slice. Okay. Now, why am I saying this? I'm saying this because, hey, I don't care about average error across the tensor, right? Of course, this will imply a bound on the average error in the tensor. I want something stronger. I want to control error slice by slice. Okay, why? Because for the applications we have, this is what's relevant. Right? So there's a stronger notion of error. I care about, this is Forbiddenious error. I care about this Forbiddenious norm error slice by slice. Okay, so hopefully none of this is controversial. But there's one thing that's controversial over here. Okay, it's the independent nature of the epsilons, or the independent nature of the missingness. And so, there you could sort of say, well, what if the missingness is sort of like some arbitrary pattern? And that's actually a very hard problem. And I'm going to come back to that later in the talk and talk about some very recent work that we've done over there that makes progress on that. That's kind of like the only kind of limiting thing in some sense in what I'm talking about over here. Okay, so great. So if we're going to do this, I need to have some sort of reasonable structure on M, right? Otherwise, there's nothing to talk about, right? So what reasonable structure am I going to assume on M? Okay, so to start with, I'm going to say something relatively benign. Okay, so what I'm going to say is, remember, slice. What is a slice? Think about CMAP. A slice corresponds here to a perturbagen. And we have cells on the rows, genes on the columns, right? So I'm going to associate some unknown latent feature, U sub C, with every cell. This is some R-dimensional vector. Okay. We're similarly going to have some R-dimensional vector associated with every gene. Let's call that V sub G. Right? You guys hopefully see where this is going, right? And now what I want to say is that any given entry, MP sub CG, right? The CGth entry of this slice MP, right, is some function of UC and VG. Okay. So I'm going to say the obvious thing. I'm going to say, hey, right, when I look at that entry, when I look at the CGth entry of MP, I'm going to ask that it be a bilinear function of UC and VG. Okay. Now, obviously, all of the complexity over here is in R, right? If R were M, then I'm making no assumptions whatsoever. Right? If the R over here were M, then this is nothing. I'm saying actually nothing. Right? There's no content in what I'm saying. And so really the whole point over here is that as you make R smaller and smaller, you're restricting the class of tensors that you actually allow. Okay. So, okay. I'm proposing this model. Why is this a reasonable model? Why should you actually be looking at this model? Okay. So I see a question from Ludwig. S is a matrix. It's a general R by R matrix. Okay. S is some general R by R matrix that's specific to slice P. Okay, so each slice P can have its own R by R matrix that identifies with this function FP. Okay, so why should you be looking at this model? Okay, why this model? Why not something else? Right? So here's the interesting thing. If you look at typical models of tensor recovery, there are two broad ways of talking about what makes a simple tensor. Okay. One is talking about the canonical rank of a tensor. Okay, so if you have a tensor with canonical rank R, that is a special case of this model. And it's a special case of this model where you will insist that every matrix SP is a diagonal matrix. All right, so let me say that again. If every matrix SP here, where I'm not going to make any restrictions on SP, I'm going to let it be a general R by R matrix. But if you ask that every SP be a diagonal matrix, this would in fact be a tensor of canonical rank R. Okay. There's another type of rank for a tensor, which is called the Tucker rank. There are others too, by the way, but these are the two dominant ones, right? So there's another one called the Tucker rank. The Tucker rank essentially places a different type of restriction on the SPs. Okay, it says that basically, if you look at these, it's a little bit more complicated, right? But it says that basically, if you look at all these SPs together, if you vectorize them, the resulting R squared by N matrix is actually rank R. If you didn't get that, don't worry about it. The point being that a low Tucker rank matrix also places restrictions on SP. Okay. So in essence, what I'm saying is, that this model is interesting because it's a looser condition on the complexity of the tensor than requiring the tensor to have either low canonical rank or low Tucker rank. Okay, so it's sort of low, it's less restrictive. I'm allowing for looser models. Okay, so in particular, if something has canonical rank R, it automatically has slice rank R. If something has Tucker rank R, it automatically has slice rank R. Okay, so simple model, right? Like super, super simple. All right. Okay, so given this simple setup, the first question I'm going to ask is what's possible? What's even possible over here, right? With a model like this. Okay, so here's what's possible. This is a lower bound. All right, so it's not clear there's an algorithm that achieves this lower bound, but this is a lower bound, right? And it says that basically the C over the capital C over here is a universal constant. What this says is that the average error that you can hope for, right, under any algorithm, so in particular the optimal algorithm, has to scale like basically R over Mn. So forget about the R square over M square that you're seeing over there. Let's ignore that for a second, right? What this is saying is the error that you can hope to see on any individual slice has to grow at least like R over Mn. Okay, let's just think about what that means for a second, all right? If you're familiar with sort of matrix recovery or matrix denoising, which I'm sure in this crowd there's a bunch of people that are, and you looked at it at recovery for a single slice of a matrix, what would you get? You get R over M. Okay, that's what you get. That's the lower bound, right? For matrix recovery, R over M. So what are we saying here? We're saying, hey, actually, right, the fact that you have all of these extra slices should help. And it helps by decreasing that error by a factor of N. Okay, so intuitively, the way I sort of think about this is sort of like, hey, you know what this is roughly saying is, let's say you have a bunch of observations of that matrix, right? They're all independent. That's one way of thinking about it. And that gives you this R over Mn. So that's a lower bound, right? R over Mn. What's the crude implication of something like this, right? Why is this interesting? This is interesting for the following reason. And this is not a precise implication, okay? So, but it's crudely how you should be thinking about it, right? So crudely, what a lower bound like this says is if you were able to pull this off and we were doing CMAP, right? Then actually the number of experiments you'd need per perturbagen, right? Remember, that's the gating factor, right? Like once you do an experiment of a perturbagen with a cell, the whole CMAP setup is such that you get gene expression for $2 on like, you know, all 1,000 positions, right? All 1,000 genes, all 1,000 landmark genes, right? It's like super cheap. And so, but setting up the experiment's expensive, right? So we don't want to do too many of these for any given perturbagen. But what this is saying is that as you sort of increase sort of the number of perturbagens, the number of experiments you need to do per perturbagen goes down pretty fast. Okay, it goes down like R over M. All right, so that's really nice, right? So that basically says that, hey, this is really good news. As I build up the CMAP library, right, by denoising in the appropriate way, I can get away with doing lesser and lesser and lesser experiments per perturbagen and still get to a good level of recovery. Okay, so the question is, can we actually accomplish this? So here's what I'm going to do. The short answer, okay, if you want to zone out, is that we can get. There's two things I want to say. Okay, first, convex algorithms. There's sort of a natural convex algorithm to actually doing recovery over here. I'm not going to talk about it, right? But it's sort of the standard thing you'd think about. Effectively fail. They fail both in theory and in practice. And I'm going to actually, and people have observed this before us, right? I'm going to present a super simple algorithm. It's kind of ridiculous. That actually comes very close to this lower bound. And this super simple algorithm in practice works well too. All right, so that's what I'm going to show you next. So let me start with the super simple algorithm. It's a really crazy simple algorithm. Okay, so what's the super simple algorithm? Okay, so remember, right? What were we saying? We were saying the representation for any slice, a slice is MP, right? The representation for any slice is U SP V transpose, right? U is like me stacking up all the latent features for the cells. V transpose is stacking up all the latent features for the genes. SP is, that's that perturbagen specific R by R matrix. Okay. So what am I going to do? I'm given this whole tensor, right? X1, X2, all the way to XP. I'm going to start with unrolling the tensor. Okay. So what's the unrolling? The first unrolling is basically take the tensor and stack it up, right? Like lengthwise, like you're sort of seeing over here. Okay. So you stack it up this way, do an SVD, followed by hard thresholding. And given SVD plus hard thresholding, so that's basically throw away all the singular values that are smaller than root n, or root, I guess, m in this case, right? Throw away all those. You're going to recover V hat. Okay, that's my estimate that I'm going to use for V hat. Okay, so I do an SVD, and it's actually, I don't even need to do the full SVD, right? I can do a partial SVD if the data set's really large, and I recover V hat by doing this. So, okay, so that's one partial SVD. Okay, here's the next one, right? Here's a second direction of unrolling this, right? Which is instead of unrolling it lengthwise, I'm now going to unroll it, horizontally. Okay, so I do that same thing. I unroll the tensor horizontally, or unfold it. That's the term typically. I unfold it horizontally. I do the same thing. I do an SVD, hard thresholding, and from that I recover the U's. Okay, I got a U hat. All right. Now, if you actually looked at this tensor stuff before, this is starting to sound like a higher order SVD. Okay, in particular, if you were doing a higher order SVD, the third step would be looking into the page. There's a third unfolding you could do. You could look at the fibers of the tensor that you get looking into the page, right? So we've stacked up this tensor, look at the tensor into the page, take those fibers and stack them up and do an SVD on that, right? And then you got a tensor product of those three things. I'm not going to do that third step. I'm just going to skip that third step, okay? So what am I going to do as my third step? My third step is really stupid, all right? So I've learned U and V, right, from the first two things. Now what I'm going to do is I'm going to go slice by slice, and I'm going to recover SP. And how am I going to recover SP? I'm going to recover it by solving that optimization problem you see on the left. If you look at this, you'll quickly realize the optimization on the left is a least squares problem. Okay, so I have a least squares problem that I solve per perturbagen, and I recover an R by R SP. Okay, what are we doing here at the end of the day, right? What we're doing here at the end of the day is we're sort of saying, look, ultimately, if you insist that the first two, the U and the V have dimension R, right? At most, right, the dimension of the third unfolding is R squared, right? And so if you insist on like being low rank in that third dimension, sure, you might actually lose something, right? Because I'm placing no restrictions on SP. But maybe going from R to R squared is not that bad when you actually have a lot of perturbagens. Okay, so if that went over your head, that's okay, right? The takeaway is that the algorithm is super simple. Hopefully you see that what the algorithm does is it does 2/3 of a higher order SVD. It doesn't do the whole higher order SVD. And for the third thing, it replaces the unfolding with essentially a slice by slice least squares problem. Okay, so very simple algorithm. You can implement it on a massive data set if you like, right? Okay, what do you get, right? So before kind of getting to the real experiments, and before showing you any like math results, right? Here's something interesting. I want you to focus on the picture on the right. Okay, forget about the picture on the left. Okay, here's the picture on the right. The picture on the right, okay, is basically a sample, a random ensemble of Tucker rank R. Okay, so Tucker rank R tensors. All right, and we did two things on those Tucker rank R tensors. One was to basically run the tightest convex relaxation for recovery on those tensors. And what you can see from that is that's the black dots, right? Is the error looks like this square root shaped curve. I do the same thing with slice learning that I just talked about, and you get this straight line. If you look at what's on the x-axis over here, the R square over M square, essentially what this is saying is up to a factor of R, this is behaving exactly like the lower bound. Okay, so the takeaway from this picture, if you didn't get the picture, that's fine. Let me make sure you get the takeaway. The takeaway is actually when you do the slice learning thing, it's within a factor of R of the lower bound. If you do something with the tightest possible convex relaxation, and there's many, by the way, The obvious convex relaxation is not correct, by the way. Okay, this is for the tightest possible convex relaxation. You actually explicitly don't get that lower bound. It's not a matter of being able to prove tighter upper bounds. Okay, so this was sort of something that was pretty exciting when we started doing this. We said, okay, can we prove something, right? And to a high level, this is what we proved, right? What we can say is, actually, if you do what I just said, the error under this algorithm, okay, so this is the Forbiddenious error per slice, right? Scales like r squared over m squared plus r squared over mn. Okay, now if you look at the thing on the left and you compare it to the lower bound, it's basically the lower bound. All right, it's instead of the leading term being r over mn, it's r squared over mn. So the scaling is correct with respect to the dimensions, which is really what we care about. Okay, you have to give up a little bit in terms of the scaling with respect to r, but you get the exact optimal scaling with respect to the ambient dimensions, which is potentially what we care about. Okay, so I'm not going to talk about how we prove this, right? But the proof's actually not that hard at the end of the day, right? The proof is, let me actually not talk about it, right? But it's not that bad at the end of the day. And there's links to this later that I'll provide if you're curious about how one establishes something like this. Okay. All right. So now let's do the fun part, right? So I've talked about this algorithm. Now what I'd like to do is actually apply it, right, to the various things we set up. So let me start with applying it. Okay. So the first place I want to apply it is the CMAP data set, right? And so coming back to the CMAP data set, let's remind ourselves what the CMAP data set was, right? We had for a bunch of different perturbagens, this cell by gene matrix. Okay. So let me actually be a little more precise this time. Okay. So what are you observing on, like, if I do, like, what's an entry of one of these matrices, right? So what's important is to say that the entry on one of these matrices, right? So let's say I take a perturbagen, I treat a particular type of cell with that perturbagen, right? I'm going to get one row of one of these slices, right? If I take a perturbagen, treat a cell with it, I'm going to get one row of one of these slices, right? So what I'll do is actually I'll do two experiments really, right? I have that row which tells me what is gene expression for that cell when I treat it with this perturbagen. What I want to do is subtract off gene expression for that cell when you treat that cell with DMSO. Why? Because we want to actually take out the first order effects of of the cell and gene expression in that cell. All right. So it's important to sort of say that really what you're measuring is kind of like a treatment effect, so to speak. All right. And we'll get back to that in a second. So what am I going to look at? Okay. So for this experiment, I restricted myself to about 1,300 odd perturbagens. Okay. And so these were perturbagens that were used on at least 10% of the cell types available. Okay, so each of these perturbagens had been used on at least 20 different types of cells. I restricted to cells that were treated with at least 0.1% of the available perturbagens. So that is to say, for any given cell that I included here, that cell had to have been involved in experiments with at least 10 to 15 odd perturbagens of this list of 1,300. Okay. And you get, anytime you do an experiment, you get the whole sort of 1,000 genes for free. Okay, there's nothing missing over there. Right? So that's the data set that we have. Okay, just to visualize the data set, right, really quickly. If I sort of, right, if I picked a column over here, if I picked a gene and I looked into the page, right? So you pick a gene, which is pick a column, and you sort of look into the page, you're going to get a long matrix, right? That's basically cell by perturbagen across, for that gene, right? What does that matrix look like for this data set? This is an example of what that matrix might actually look like for one specific gene, okay? But because the gene, you get all the genes, this is basically the missingness pattern for all genes, right? And so what do you see from this missingness pattern, right? What you sort of see from this missingness pattern is it's very, very sparse, right? Close to 80% of the experiments, so to speak, are missing, right? Like of the potential experiments we could do, we've done like 20% of them. And we want to impute like the rest of the 80%, right? That's sort of effectively the picture over here. Okay, so let's see how well we can do this. All right. So what are we trying to estimate? What we're trying to estimate, as I said, is basically what's the effect, what's the incremental effect, right, of treating a particular cell with a particular perturbagen relative to treating that cell with DMSO, right? So that's really what's in every one of these matrices. Okay, so it's important that you do that, right? Otherwise, the problem gets trivial, right? So that's what I want to measure. So let's see how well you can do this, right? So the first thing we'll do, okay, is this simple thing from this paper. There's a couple of papers that do this. And these papers point out that the problem that we've set out to describe is actually remarkably hard. Okay. In fact, when these folks wrote this paper, their point was that you actually get, because by the way, prior to these papers, there was a lot of kind of junk stuff that had been done for imputation. And these guys pointed out that actually, if you just account for perturbagen-specific fixed effects and cell-specific fixed effects, you already get basically very close to state-of-the-art performance. Okay, and the state-of-the-art was roughly this 0.23 at the time, right? Not great, by the way, right? In and of itself, it's terrible, right? And what these guys said was actually, if I just look at cell-specific effects and perturbagen-specific effects, you already get that. All right, this is very disturbing, right? It's disturbing because what you care about biologically is the interaction effect, right? You care about the interaction between the perturbagen and the cell. And in fact, you care about the interactive effect, hopefully in a way that's gene specific, right? And so this is kind of a little troubling. Now, last year, there's this really nice paper by Carolyn and Devavrat and others, okay, that said, hey, let's do this idea of basically synthetic control, right, applied to this problem. And actually doing this allows you to capture this interactive effect. When you do this, you get an R square of 0.25. So it's an improvement over the 0.23 because you're allowing interactive effects. If you do what I just talked about, the R square basically doubles. Okay, so you get, just using the vanilla slice learning thing I just described, you get an R square of 0.4, which is a massive leap from where this was. The last line, which we're calling slice plus neural net, is basically where I take that last step where you're learning, where you're doing that projection, where you're solving that least squares problem, and replacing it with simply learning a neural network. Okay, some neural network of these latent features that were learned early. Okay, so point being that if you look at this, it works really well. Picture is worth 1,000 words. So what I want to show you next is actually, if you dig in, right, does this actually impute well, right? And so here's a pictorial way of doing that. Okay, so I'm looking at a specific cell over here. Okay, this is a skin cancer cell, right? And I'm going to look at a very specific perturbagen. Okay, I'm not even going to try and pronounce this. I don't know what this means chemically, right? Something. It's the descriptor over here is basically a mechanism of action, right? It's telling you how this works. Okay, so I take the cell, I treat it with this perturbagen, and we have ground truth. Okay, so you know exactly the gene expression you got. And what I've done is, because the gene expression is standardized, I've just organized these genes from least expressed to most expressed. Okay, so from left to right, blue is least expressed, red is sort of most expressed. Right? That's really what we're seeing, and that's ground truth. Now, just to kind of get a sense of things, let's say I randomly predicted something. I ordered this randomly, right? Just to get a color picture, right? You get something that looks like this. And so the natural question is, if I now look at the predictions we're making, is it closer to random? Is it closer to the thing at the top, right? And if we're doing a good thing entry by entry, hopefully it looks like the thing at the top. And indeed, you get a picture that looks roughly like that, right? What you see is like what's predicted, that's in the middle. What this pictorially shows, more than what the R square was showing on the earlier slide, is really what you're getting, as you do this imputation. Okay, so that's CMAP. Second example. Okay, so here's another example, and this was sort of the thing about recovering ubiquitination, right? So the data set actually looks something like this. right? But there's not 3 slices, there's actually 5 odd slices. And right enough, right, the ubiquitization slice is the hardest slice to impute. It has the highest amount of missingness, right? And in fact, its missingness is such that entire samples are actually missing, right, from that data. So what we're going to try and do is run this process to effectively predict ubiquitization at a specific site on a specific sample, right? And so that's kind of, in a sense, a classification problem. And what we find is that when there's some data available on the sample, you get a very high AUC of 0.91. Remember, this is site-specific, okay? So a site-specific AUC of 0.91. If the entire sample were missing, right? So I had no information on that sample, right? By using omic data from other channels on that same sample, I'm able to speak to ubiquitization with a pretty high AUC, an AUC of 0.8, which, by the way, is very surprising, okay? Because biologically, like, getting ubiquitization data is very hard. Okay, so the MS thing is, the mass spec is very noisy in collecting this. And so being able to do this like this is actually, it actually helps a ton. All right, okay. Let me end with sort of talking about non-random missingness, right? So for everything I was talking about so far, the missing patterns for the theory, not for the practice of it, but for the theory of it, was sort of IID. For a rigorous sort of causal analysis, what you want is sort of non-random missingness. We want the missingness patterns to be completely non-random. What we've been able to do over there, okay, is essentially solve that problem for a certain special case. I'm not going to sort of really belabor the special case over here, but it suffices to say that while it's a special case for biology, it turns out to substantially generalize a setting people consider in econometrics, and in particular within something called synthetic control. So over there, we came up with the first optimal algorithm. I'm not going to talk about it over here, but this was actually an oral presentation at NeurIPS in December. And so there's a video available because of the oral at NeurIPS. And so if this is something you're curious about, you can just check out that video. And so just to end, right, what are the relevant papers in case you're curious about them? The actual mechanics of the algorithm I'm talking about are something we worked on actually 1.5 years, 2 years ago. Okay, it's a very simple algorithm. The first version of this was published in MS in 2019. The applications I didn't talk about, okay, which is these are the sensors, right? These are the biological sensors where we applied the same thing. We applied this, as I said, to lung cancer. That's the Nature Communications paper in 2020. Last year, we applied it to Alzheimer's. So early detection of Alzheimer's in a non-invasive way. That's in Advanced Healthcare Materials from last year. And if you're curious, as I said, about this sort of thing, the theory around non-random missingness, that paper was in NeurIPS in December. In many ways, by the way, it's that last paper that's the subject of like really what the current J-Clinic whatever grant was about. But it's pointless talking about that without actually talking about why it's important. And so so that's kind of, if you're curious about work around this, that's this. And with that, let me stop.
Thank you so much, Vivek. That was inspiring. I love it. That was a great talk. Thank you. And I have an immediate question related, and maybe the audience has also some questions, but related to the work. Here we're talking always about prospective data or retrospective data on the work you've been doing, for example, with lung.
Yeah, so the lung cancer work was actually a prospective study. So we designed it. Yeah. So we designed the story with that paper, we submitted it to Nature Communications, and they didn't like it because the study was retrospective. Then we designed a prospective study. The prospective study had 1,000 samples, and so it was a large prospective study. It had high power. And so it's from a prospective study.
I see. And you envisioned and then this could be applied to other pathologies as well?
Yeah. So in fact, the stuff from these two papers, the two papers in the middle, the Nature Com paper and the Advanced Healthcare Materials paper, device we built a device now that does this, and the device is now actually broadly available. And there's a number of labs that are actually experimenting with the device and experimenting with this data. So Oregon State's doing it. Steve Carr is doing it in the Broad, and a bunch of other folks. And yeah, it doesn't have, it's the purpose is doing, sort of high throughput proteomics, yeah, as opposed to anything else. So you could apply it to anything.
Excellent. And I think we have a question from Peter. He's asking here in the chat if new interactions between genes and perturbagens were discovered or even rediscovered.
Yeah, indeed, yes. So I actually cut those slides out. But one of the interesting things over here, right, is you get a really nice representation of the perturbagen. And given that representation of the perturbagen, you can now try and ask, right? If I'm looking for, let's consider the inverse question, right? The inverse question, which is really what this is about. If I'm shooting for a particular expression, right, like I'm trying to kind of like deregulate something, for instance, right? That's pushing for a particular type of expression. Now I can actually look, instead of like hunting blindly, I can literally write an optimization problem in terms of the learned SPs to say, this is actually the set of SPs I might actually want to look at. So that's actually a great question. I didn't talk about it because it is too much to talk about, but yes.
Yeah, thank you, Peter, for the question. Yes, we still have maybe a couple of minutes. If anyone, yeah, and Peter said thank you. If there's any other question, but that's great. You guys have been doing great work here, and yeah, maybe this Jameel Clinic grant can also benefit from some of this work. So I hope that, like, how is the work coming, by the way?
It's good. I mean, this is where we're at, right? I talked about where we're at. So, I'm pretty happy with it. I think on the proteomic side, I think there's some new stuff we're kicking off where I think there's, at a high level, I think there's a lot of opportunity to kind of question fundamental algorithms people actually use to interpret the raw data that comes out of mass spec. So ultimately, if you think about all of the proteomics, it's all driven at the end of the day by mass spec. And right, like, so you can do all these clever sensors, but at the end of the day, you're still driving it with mass spec. And so now the question becomes, okay, great, if you're driving with mass spec, what can we do with the mass spec, right? Mm-hmm. And I actually think that this is shockingly, like, for whatever reason, like a lot of like the deep learning tools we have not actually been applied there. And in particular, I think there's sort of like a variety of like generative modeling that can help, I think, invent a new generation of sort of mass spec inference algorithms. And this is important because this is a bottleneck for the entire pipeline.
Yeah. Excellent. Let's see if we have maybe another minute, if any other question. But yeah, thank you very much.
Awesome.
So
hey, thank you.
We'll let it here. Thank you again, Vivek. And if anyone is interested on this work, there's some reference here. We're going to be able also to have the recordings online. There's always a way to follow up with the faculty or with Jameel Clinic. You know where to find us. Thank you again, everyone, for joining us today. And thank you, Vivek. Take care. Talk to you very soon.